Hello friends, you are welcome again in our video lecture series on the topic Bryophytes. So today I am going to discuss the classification of Bryophytes as per the classification system made by this G.M. Smith in 1955. So according to classification of G.M. Smith, the division Bryophyta is divided into three classes. The names given by this G.M. Smith to these classes are Hepatici, Anthocerotes and Muci. So afterwards, in 1957, the worker Proskauer in 1957, he modifies the system of classification. That means he only modifies the names. He suggested the different names for the classes like Hepatici, Anthocerotes and Muci. So according to or by following the ICBN rules, ICBN means the International Code of Botanical Nomenclature rules, this worker Proskauer in 1957 replace the terms or names of the classes Hepatici with the word Hepaticopsida. The Anthocerotes word is replaced by Anthoceratopsida and word Muci is replaced by Bryopsida. So according to International Code of Botanical Nomenclature, this Proskauer replaced these terms and give the names for these classes like Hepaticopsida, Anthoceratopsida and Bryopsida for the three major classes of division Bryophyta. Then this Hepaticopsida, generally the members which are included in this Hepaticopsida are commonly known as liverworts because the shape of the thallus or gametophyte is resembling the liver in our body. That's why the members of this class are commonly known as liverworts. Anthoceratopsida, commonly the members of this class, Hepaticopsida members, are generally known as hornworts because the structure of the sporophyte in this Hepaticopsida is as like the horns of the bulls. That's why the members in this class, Hepaticopsida, are commonly known as hornworts. And finally, this last class, Bryopsida. The Bryopsida class, these are including the mosses. That means as far as the different Bryophyta members are concerned, Bryopsida class members are supposed to be advanced type of Bryophytes. That means these are showing differentiation of plant organs like rhizoids, which are multicellular, stem axis as well as leaves, as well as another important character of this Bryopsida. These are commonly known as leafy gametophytes. Sporophyte in this class is well differentiated into food, seta and capsule, these three distinct parts. So as compared to this Hepaticopsida, Anthoceratopsida, the members of this Bryopsida includes all types of the mosses. Then this Hepaticopsida class is divided into four orders, namely Spherocarpales, Marchantiales, Jungermannials and Calobrials. This second class, Anthoceratopsida, is having only one order known as Anthocerotes. This third class, Bryopsida, is divisible into three subclasses like Spinobria, Enterobria and Eubria. This Spinobria subclass is having only one order known as Pagnales. This Enterobria subclass is having only one order known as Andrians, and this last subclass of Bryopsida known as Eubria. This Eubria subclass is largest class and it is having 14 orders, 14 different orders. So this is the classification of the Bryophytes up to the orders as per our syllabus made by the G.M. Smith and modified by Proskauer in 1957. So I would like to discuss in short, the few characters of each class: Hepaticopsida, Anthocerotopsida, and Bryopsida. So I have told you earlier, the first class of Bryophytes, known as Hepaticopsida. Commonly, the members of the Hepaticopsida are commonly called as the liverworts because of shape of this thallus or gametophyte is as like liver in our body. That's why they are members of this class are commonly known as liverworts. So the plant body in this Hepaticopsida members is dorsiventrally flattened. Plant body is very simple. All the members are not thalloid forms. Some forms are thalloid, which shows dorsiventrally flattened body, while some forms are showing the lippy gametophyte. That means lippy gametophytes as well as thalloid gametophytes are observed in this class. This is another character of this, important character of this class, Hepaticopsida or liverworts. Then, if we observe the ventral side of this thallus of Hepaticopsida members, the ventral side of thallus occupies the unicellular rhizoids. The rhizoids are of two types: tuberculated rhizoids and smooth walled rhizoids. For example, in Ixia. In another members, rhizoids are unicellular, no doubt. That means unicellular rhizoids and scales, multicellular scales, they are present on the ventral side of this thallus. This is another character of this class, presence of rhizoids and scales on the ventral side of the thallus or gametophyte. Then, after maturity, the gametophyte develops or produces the gametes or sex organs. Sex organs are generally produced on the dorsal side of the thallus. For example, in Ixia, on the mid-dorsal line, the male gametes producing structure known as antheridia, female sex organs known as archigonia. Both sex organs are endogenously developed inside the thallus tissue on the mid-dorsal line, while in some members the reproductive structures are present at the terminal position of the thallus. For example, in Markensia, reproductive structures are not found inside the thallus, they are present on the terminal position of the thallus. For example, in Markensia. Next character about sporophyte. This sporophyte in this class, in some members, for example, Ixia, sporophyte is very simple. It is represented by only capsule. Foot and seta, these two regions are totally absent in lower bryophytes like Ixia, in Hepaticopsida members, while in some forms, for example in Markensia, sporophyte is divisible into foot, seta and capsule, these three distinct regions. Then sporogenous cells are developed from endothecium region, that means inner layer of the sporophyte. Sporogenous cells are developed. This capsule region do not contain columella in this Hepaticopsida class. So these are the important character of the Hepaticopsida. Then if we observe the important characters of the Anthocerotopsida, so all members in this class Anthocerotopsida, they are generally known as liverworts because the nature of sporophyte in these Anthocerotopsida members is as like the horn shape. That's why they are commonly known as hornworts. So gametophyte of these hornworts or Anthocerotopsida members is also dorsoventrally flattened. It is generally lobed. Then rhizoids are only one type. Smooth walled rhizoids are only observed in this class. Scales are totally absent. Only smooth walled rhizoids are there. Scales are absent. There is no internal tissue differentiation. As far as if we observe the transverse section of thallus or internal organization of thallus, thallus is homogeneous. That means it is made up of same kinds of cells. All cells are parenchymatous cells containing chloroplast. Chloroplast. That means there is no tissue differentiation. Thallus is homogeneous. It is made up of one type of cell. One type of cell. Then, if we observe the reproductive structures, Anthidea and Archigonia, these reproductive structures are superficially present on the dorsal side of the thallus. They are also endogenous. That means internally, inside the thallus, on the dorsal side of the thallus, Anthidea, Archigonia, these male and female reproductive structures are developed. As far as sporophyte is concerned, at the end of gametophytic stage, sporophytic stage begins and sporophyte shows the horn-like structure. Sporophyte is developed on gametophyte. It is not independent. It depends on gametophyte. That means sporophyte is divisible into foot, seta and capsule. Capsule region is lacking the columella or sterile portion in this class, Hepaticopsida. If we observe the characters of this last class, Bryopsida, the Bryopsida members, the main plant body is a gametophyte, and this gametophyte shows the two stages. The first stage in the gametophyte is commonly known as protonemal stage, and second stage is commonly known as leafy stage. That means there are two stages reported or observed in the gametophyte. First stage is known as protonemal stage. It is juvenile stage, younger stage. The protonemal stage is prostate one. It is extensually branched one. It is green one. It is heterotropic one. Later on, leafy stage shows the erect cylindrical stem axis and small simple sessile leaves. Leaves in this Bryopsida member are generally sessile one, simple one, showing single midrib. Lateral veins are totally absent. If we observe the rhizoids in this Bryopsida are multicellular. Rhizoids are multicellular. In Anthoceratopsida and Hepaticopsida, rhizoids are unicellular. While rhizoids in these Bryopsida members are multicellular, the sex organs are developed in groups and the apical regions of the branches. That means anthedial head contains Anthidea, archigonial head contains Archigonia. That means sex organs, reproductive structures in the form of Anthidea and Archigonia, they are developed in groups at the apical regions of the fertile branches. Then, if we observe the structure of Anthidium is a club-shaped structure. It is containing the many antherozoids, actual male sex cells. After maturity, it diases and antherozoids are liberated. Archigonium is a flask-shaped structure. It is having short stalk, narrow apical neck region and basal bulbous venter region containing venter canal cell and egg cell containing egg nucleus. So, this sporophyte in this Bryopsida member is well advanced and it is divisible into three distinct regions like foot, seta and capsule. So, this sporophyte after maturity produces the spores, and in this sporophyte there is a sterile portion in the form of columella. So, presence of columella or sterile region is the important character of sporophyte. So, this sporophyte liberates after maturity of this sporophyte. Afterwards, this spore germinates when gets suitable condition for germination and gametophytic stage begins. So, that's all about the classification of this Bryophyte. Then I am going to discuss in short the alternation of generation in Bryophytes. So, all Bryophyte members shows this important character, alternation of generation. So, main plant body of all Bryophyte members is a gametophyte. So, this gametophyte is generally haploid. So, this gametophytic stage, haploid stage, is the main plant body. This Bryophyte members reproduces by vegetative method as well as by sexual method. When Bryophyte thallus becomes mature, it produces the sex organs. The sex organs are developed in each and every bryophyte member. The male sex organ which is developed in the bryophyte is commonly known as antheridium. Female sex organ which is developed in bryophyte is commonly known as archegonium. This antheridium is club-shaped structure. Antheridium contains many androgonial cells or antheridium mother cells. Each antheridium mother cell after meiosis produces the antherozoids. Female reproductive structure is archegonium. It is a flask-shaped structure having narrow neck region and basal bulbous fender region containing fender canal cell and egg cell. This egg is the actual female sex cell. Antherozoid is the actual male sex cell or male gamete. So this antheridium and archegonium field produces egg and antherozoids. With the help of water drops, the antherozoids are transferred from this antheridium towards the archegonium, and antherozoid unite with the egg. Union of the antherozoid with egg is commonly known as fertilization. So product of fertilization is the zygote. So after the fertilization, gametophytic stage shows haploid condition. After fertilization, diploid zygote is formed. From this zygote, there is a formation of oospore or sporophyte. So this zygote is the beginning of formation of zygote is the beginning of sporophytic generation. This is the diploid stage in its life cycle. So this sporophyte in all bryophyte is not independent. Sporophyte always depends on gametophyte for getting its food material or for getting nourishment. That means sporophyte develops on the gametophyte. It is not independent structure. Sporophyte depends on gametophyte. Field sporophyte develops on gametophyte. This sporophyte produces the spore mother cells. The spore mother cells undergoes meiosis, and after meiosis, haploid spores are produced. That means formation of spore is the beginning of gametophytic generation. That means the gametophytic generation is produced from the spore after germination of spore. That means in the single life span, each and every member of bryophyte shows two stages. That means gametophytic stage and sporophytic stage. That means gametophytic stage alternates with sporophytic stage. So if we observe this alternation of generation, there is a alternation of generation in this bryophyte members is heteromorphic. What is the meaning of the term heteromorphic? That means morphologically the gametophytic stage, male plant body bryophyte is gametophyte. It is haploid. Morphologically, this gametophyte is totally different from this diploid sporophyte. That's why alternation of generation in bryophyte is commonly called as a heteromorphic alternation of generation. That means alternation of generation is the very important character in each and every bryophyte. So that's all about the today's two articles, classification of bryophytes as per the G.M. Smith, and next article, alternation of generation in bryophytes.